Kogelberg Biosphere Reserve in the Cape Flower Kingdom is one of the world's most recognized biodiversity hotspots. It actually boasts the world's highest plant species biodiversity per unit area. The CSIR initiated a project where we wanted to use bioprospecting techniques to extract value from microbes in the soil at Kogelberg Biosphere Reserve. Knowing that there's high plant biodiversity, this could translate very directly to microbial biodiversity. In our case, we believe that the high diversity would result in new genes for making protein molecular biology reagents. These protein and enzyme reagents are used widely all over the world by researchers to conduct research. In our case, we are targeting specifically molecular biology enzymes, which are used to cut DNA. In some instances, they're also used to amplify and make more molecules of DNA, and in other instances, they're used to, in fact, join DNA molecules. These molecular biology techniques that utilize these reagents are a crucial component of the genome editing and genome engineering revolution going on at the moment. We believe that the genes and reagents we find from the soil at Kogelberg will form a key component of commercializable research coming from the CSIR. The work is funded by the Department of Science and Technology, and through their generous contribution, we have now made excellent progress in the lab. At the moment, we have been able to sequence the full metagenome from this soil and extract gene sequences that are ready for expression. We now have several processes for manufacture of these enzymes ready in the lab to translate into functional product prototypes. Several of these protein reagents we've isolated from Kogelberg are now entering a phase of end-user testing, what we call beta testing, where end users and potential customers are testing them for use in their own applications in the lab. The next steps will then be to establish if in fact the end user testing has been successful, to facilitate then a strong case for translating this into a commercializable prospect for the CSIR. We see this as a real opportunity for generating new SMMEs, for extracting value from South African biodiversity, and we are hopeful that in the next couple of years there could be an entity that will commercialize these enzymes into real value for the country.